This is our project, Antibacterial Defense of Different Spices.
Our procedure was or had a sample size of 2 petri dishes, 1 swab of E. coli, and a half teaspoon of spices. Our test subject were 5 spices, which were oregano, thyme, cinnamon, onion powder, and salt. Our independent variable were the spices, and the dependent variable were the size of the inhibition zone in millimeters. The constants were the petri dishes, the amount of each spice, the time given for each spice to take effect, and the attempted even spread of bacteria. Our control was the petri dish with E. coli and no spice. We had 2 replicates and a frequency of recording after 48 hours from when the spices are added. And our statistical analysis was standard deviation and standard error. And each person would carry out 1 trial of each spice to split the work.
Okay, and for the safety procedure, we planned out how to safely deal with the bacteria that we used in the experiment. We will safely wear wear safety gloves and put on goggles while handling the bacteria. We will make sure to streak the plate evenly and not get the bacteria anywhere else, as well as making sure to not touch the plate directly or open to the lids once the E. coli has been added. To dispose of the swab and petri dish, we will disinfect it with isopropanol. Making sure to wear gloves and not touch the plates directly, we will pour in the isopropanol into the plates and let them sit for 15 minutes before gently disposing in the trash. This will kill off and sterilize the bacteria, allowing for disposal in the trash.
Our hypothesis was if oregano, thyme, cinnamon, onion powder, and salt were to be put in petri dishes with E. coli spread in them for 48 hours, then cinnamon will create the largest inhibition zone because it contains essential oils that counter E. coli and many other bacteria.
Here are some images of the experiment with replicates after 24 hours and another separate replicate from the other partner with after 24 hours.
Here are images after 48 hours when recording with onion powder, cinnamon, thyme, and oregano. As you can see, the thyme and oregano both had the spice bleeding into the E. coli and didn't create much of an inhibition zone, while the onion powder and cinnamon had a noticeable one.
This is our graph for the experiment. Um, After the 48 hours, it was found that there was a statistical insignificance between the cinnamon and the onion powder. Additionally, the thyme, oregano, and salt also showed to have a statistical insignificance shown by the error bars on the graph. On average, the onion powder performed 12 millimeters better than the thyme and 12.5 millimeters better than the oregano and the salt. The cinnamon also performed, on average, 12.5 millimeters better than the thyme and 13 millimeters better than both the salt and oregano.
Okay, those are table and annotation. After 48 hours, the cinnamon and the onion powder showed to be statistically significant from the thyme, oregano, and salt. The spice that contained the least variations in the data was the onion powder, with an average of 0.5 millimeters. The spice that contained the largest variations were the cinnamon, with an average of 2 millimeters. On average, the cinnamon performed 1.5 millimeters worse than the onion powder, 1 millimeter worse than the oregano and salt, and zero point 0.5 millimeters worse than the thyme. The onion powder performed an average of 1.5 millimeters better than the cinnamon, zero point 0.5 millimeters better than the oregano and salt, and 1 millimeter better than the thyme.
And this is our conclusion. In conclusion, both the cinnamon and onion powder performed the best with no statistical significance between each other. However, the onion powder did contain the smallest amount of variation in data. The other three spices performed poorly and were statistically significant from the onion powder and cinnamon, but not to each other.